coconut, buffer, measuring cylinder, filter cloth, pH indicator, bottle, papaya. Day 1.
We first cut unripe papayas into thin slices and then we measure out 100 grams. We then measure out 100 milliliters of buffer solution.
And then we blend them until smooth.
We then strain the papaya juice. We then test the pH. And as we can see, it is approximately 6.
Then we pour the papain juice to a clean bottle.
Now it's time to make the coconut milk. To make the coconut milk, we first put grated coconut and coconut water 1 kilogram each into a big basin. Then we squeeze the grated coconut, and then this process will create the coconut milk. We then extract the coconut milk from the mixture by using sieve and squeezing the coconut milk into a big clean container. We then pour the papain juice into the coconut milk. We can swirl the mixture until it is thoroughly mixed. After it's thoroughly mixed, we can then close the lid, and then we can store them in a cool place, not under direct sunlight.
Day 2.
This process causes fermentation in the mixture, which may cause gas buildup and may open the lid. This fermentation process caused the mixture to split into many parts, the lowest part being the grated coconut, the middle part being water, the second top part being oil, and the topmost part is the coconut waste called the blondo. We then measure out the sodium hydroxide to make a 0.1 molality solution. We use 1 gram of sodium hydroxide to 250 milliliters of water, and then we stir them until the solution is homogeneous to create the 0.1 molality sodium hydroxide solution. We then try to separate the oil from the blondo through a filter. We use many objects like spoon, needleless syringe, even the cloth filter we use for the papain juice. Because we didn't use a syringe, we let the blondo oil mixture to sit so it separates again, and then we use pipette to separate the oil. We then pour the oil into a clean bottle, and then we centrifuge it for approximately 2 hours.
Day 3.
It is now time for the organoleptic. We need to prepare the sodium hydroxide, the 70% alcohol, and the PP indicator. We first measure out the oil we have. And then we test the pH of the oil first. As we can see, the pH is approximately 5. We then measure out 25 milliliters of 70% alcohol, and then we pour 10 milliliters of the oil into an Erlenmeyer flask. We then pour the alcohol also into the Erlenmeyer flask, and then we swirl them together. And then we put 1 milliliters of PP indicator into the oil alcohol solution we just made. And then we swirl it around again. We then titrate the sodium hydroxide until it changes color, and then we slowly swirl them around until the color reverts back. We can then test the pH of the mixture we just titrated, and the pH is approximately 7.